This
Disturbance.
conference will now be recorded.
Okay. So as we started with the cell, right? So cell. So as we know that cell is the what? Cell is the structural and functional unit of life. It means what? It means all the organisms on the earth, they are being structured because of cell, and all the functions they do, all the necessary function for the survival they do, they are being performed by the cell. So that's why cell is the structural and functional unit of life. So as we know that the first cell formed, very first cell formed, where it is formed, anybody have any idea? You can respond me in chat box. So where the very first cell has formed? Yes? So the very first cell has formed in sea, or we say in ocean. So the life originated in ocean, and that's why the if you observe closely, the composition of cell and the composition of ocean is match. It's somewhat similar. Okay, so the very first cell is formed in ocean, and the very first cell formed was very simple type. Okay, very simple type. It was consist of just genetic material within and a waxy oily lipid layer. So very first cell formed was consist of a membrane and the genetic material inside, that is DNA. And further development of the cell according to the environment, as the cell wanted to adapt the environment. See, the environment of earth is dynamic, is constantly changing, and cell has to do adaptation according to environment for the survival. If they will not, they will get distinct, right? As dinosaur died, like that. So cell has improved itself into many aspects. So on the basis of the development cell has did in it, so we have now on the earth two type of cells, right? We have two types of cells on the earth on the basis of their development. First we have prokaryotic cell, and another we have eukaryotic cell. So what is the basic difference between prokaryotic cell or eukaryotic cell? So the prokaryotic cells are the very simple type of cells which lack true nucleus, which lack true nucleus. So inside this cell, there is the cell, they are consist of cell membrane. Inside them there is cytoplasm and the genetic material, which is we called nucleoid, which we called nucleoid, right? So there is no definite nucleus within this cell. The nuclear material is just kept inside the cytoplasm and adhered to the membrane, or freely suspended. There is no protection to genetic membrane, right? But as cell realized that genetic material is so very important to pass on the information to the next generation, right? So all the information, what genetic material contains? Genetic material contains all the codes for protein formation. What genetic material contains? All the codes for protein formation. And proteins are the primary functional units of all the processes, right? Almost all the processes inside the cell, they are being performed with the help of protein. They act as enzymes. So genetic material is very important. So cell has developed a protective membrane around the genetic material. Cell has developed a protective membrane around the genetic material, right? To protect it. So that membrane, or the so that compartment we called what? That compartment we called nucleus, right? That compartment we called nucleus. And with this, again, cell has realized that many of the processes that should be separately performed. So, cell started forming various compartments within the cells that we called organelles, right? So prokaryotic cell is a very simple type of cell lacking true nucleus, and there are no organelles, right? No organelles. Organelles means what? Organelles are nothing but the compartment in within the cell for the specialized functions. So the prokaryotic cell, it only has ribosome, right? It lacks all other organelles except ribosome. And eukaryotic cell is consist of various cellular compartments called organelles like endoplasmic reticulum, Golgi apparatus, lysosomes, right? Mitochondria, chloroplast, and so on. So, this is the basic difference between the prokaryotic cell and eukaryotic cell, right? A cell with true nucleus is eukaryotic, and a cell with primitive type of nucleus, not well developed nucleus, is the prokaryotic cell, right? So, these words prokaryotic and karyotic, right? So carry-on means nucleus. Carry-on mean nucleus. And pro means primitive. Pro means primitive. So primitive means old, right? So these prokaryotic cell, they doesn't have true nucleus. They are the old type of cells. And eukaryotic cells, you means developed. You means developed. So they contains well developed nucleus, that's why they are called as eukaryotes, right? So we know that as we classify all the living organisms, hope everyone is making notes as well, right? And if you have any doubts, you can ask me anytime in the chat box. So let's go ahead. So as we know that all the living organisms, right? All the living organisms on the earth, we have classified them into five classes, right? We have classified them into five classes to make our study easier about the all the living organism, and that classification system which is introduced by the Whitaker scientist. Whitaker is called as five kingdom classification, right? So according to the five kingdom classification, we have classified organism into five classes. First is Monera, second one is Protist, or we say Protista, third one is Fungi, fourth is Plantae, and the fifth one is Animalia, right? So among these all five classes, the very first class Monera, it includes the prokaryotic organisms. It includes the prokaryotic organisms, and all other four classes, they includes the eukaryotic organisms. Okay, this much clear, everyone? May I have some responses in chat box? Are you all getting? Yes? Great. Good. Chalo. So now, one more thing we need to understand here is first, on the basis of the cell type, we have classified organism as prokaryotes and eukaryotes, right? And again, on the basis of the number of cells they have, right? So next we classify organism on the basis of number of cells organism have. So on the basis of this, we can classify organisms as unicellular and multicellular organisms, right? Unicellular and multicellular organism. So all the organisms which are consist of single cell, right? What is mean by uni? Single, right? And unicellular means having single cell only. So all the life processes required for survival are being performed by only single cell. Everything, the respiration, excretion, metabolism, all the things, they are performed by a single cell. Such organisms, they are called as unicellular organism. And the multicellular organism are the organism which are made up of more than one cell, right? So that are called as multicellular organism. So here when we classify all the organisms in this five classes, so the very first class, Monera, it includes all the unicellular organisms only. Okay. So can we say in this first class, Monera, there are all unicellular prokaryotic organisms are present, right? And if we discuss about the protist, so protist also contains unicellular organism, but they are of eukaryotic type, right? So the protist, they contains unicellular eukaryotic organisms, right? And the Monera, they contains unicellular prokaryotic organism. Okay. And if as we go further, the fungi, again see the fungi, plantae, animalia, all will be consisting of eukaryotic cell type. And from fungi we can form the group fungi includes some unicellular and multicellular as well. Okay, so you can see as we are proceeding further with the classes, the features are increasing. Now, the next plantae, they consist of all multicellular organisms with some characteristic feature. Again, animalia, they also consist of multicellular organism. So now we have clear idea that what are cells, and all the living organisms, they are made up of cells, types of cells, and on the basis of types of cells and number of cells, we are grouping the organism into five classes, right? So all these five class of organism, they have the genetic material, right? Because genetic material is responsible to transfer the characters to the next generation. So all these organism, they possesses genetic material as DNA. So only exception to this are viruses. Viruses, they are grouped into the different class. They are not even included in this five kingdom classification, right? Because viruses, they are the connecting link between the living and non-living. So despite of this five kingdoms, we have a separate class in which we have kept some organism which are not exactly living or non-living, that is viruses. And they have one more exception, that some of the viruses, they can have RNA as genetic material, okay, as well as they can have DNA. So except viruses, all the organisms on the earth, they are possessing DNA as genetic material. Okay, so now we have came to the our today's actual topic we want to learn, that is chromatin structure, right? So what is chromatin? Chromatin is nothing but the condensed form of DNA. What is chromatin? So chromatin is nothing but the condensed form of DNA. So condensed form of DNA is chromatin. So before going to start with the chromatin structure, we need to first understand what is DNA. So can anybody, so many of you can respond the full forms of DNA and RNA in the chat box. So let us discuss now about the DNA. Everyone is getting? Yes? Okay, good. It is deoxyribonucleic acid. Good. Okay, great. And what about RNA? Very nice. Ribonucleic acid and deoxyribonucleic acid. So let us study the structure of DNA in detail, and on the structure of DNA you will be getting questions in the exam. Okay, many a times they have been asked the questions on this topic, structure of DNA. So let us start discussing the structure of DNA. Good, everyone. Nice, right? Chalo. So let's go ahead and talk about the structure of DNA. Because now we know that the DNA is the genetic material, right? Structure of DNA, that is deoxyribonucleic acid. So before starting with the structure of DNA, first let us discuss the biomolecules. So what are biomolecules? So all the living organism, they consist of cell, and cell consist of some major molecules. Cell is consist of some major molecules. The molecules which are responsible for forming cell, or which are responsible for functioning in living organism, they are called as biomolecules, right? So the biomolecule, this word is comprising of two words, right? Bio means living, and the molecules, right? We know that molecules are nothing but the combination of two or more atoms, right? That is two or more atoms of same or different type, right? Molecules are what? They are the combination of two or more atoms of same or different types, right? So all the molecules which are working or which are functioning in biology in living systems, they are called as biomolecules. So we have four major biomolecules working in the living organism, and that four major biomolecules, the very first is carbohydrate. Make a note of this, everyone. You are going to study all this topic in the biochemistry in detail. Okay, so here we are going to talk about DNA. So first very important biomolecule is carbohydrate. The second one is proteins. Then we have lipids and nucleic acid. So these four. Despite of this, we have vitamins, right? And other molecules as well. But these are the four major biomolecules. Okay. And today we are going to talk about the nucleic acid. So nucleic acids, they are of two types. So in the living cells, we can find the two types of nucleic acid. The first one is DNA, and the second one is RNA. So as you all have mentioned, DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Okay. So the DNA is going to act as genetic material, and the RNA, it has various cellular function, right? So RNA has various important cellular functions in all the living organisms, right? Except we say viruses, right? Except viruses, all the organisms, they have DNA as genetic material. So now let us talk about the DNA structure. Structure of DNA. So the DNA is mostly a double helical structure. What is DNA? DNA is a double helical structure. It means it consists of two strands. DNA is consist of two strands. Okay. So if we see, these are the two strands. Okay. So DNA is consist of two strands, and these two strands, they are wounded around a central imaginary axis. So if we want this two strand on an imaginary axis, a central imaginary axis, so they will form a helical structure, right? This is an helical structure of DNA. So DNA, that's why it is called as double helical structure. So DNA consists of, or these two strands of DNA, right? So these two strands of DNA, they are consist of some smaller units, right? They are consist of some smaller units. So DNA, it's an polymer. DNA, it's a polymer. What is mean by polymer? Polymer means anything which is consist of small units. है ना? Polymer is formed by joining monomers. What is monomer? Monomer means single unit. What is mean by monomer? Single units. So this DNA is also consist of monomers, right? It's an polymer, so definitely it is going to have some monomer, the single units. So the DNA is actually the double helical structure. It's a polymer of, so DNA is polymer of nucleotides. What I'm saying, the DNA is polymer of nucleotides. Okay, so now let us discuss the structure of nucleotides. As we got to know that both of these two strands, they are consist of nucleotides. By joining so many nucleotides together, we have got the two strands, and these two strands we have interwounded, right? These two strands we have interwounded to get the double helical structure. So now we get to know that nucleotide is the very basic unit of DNA. So now we will discuss the structure of nucleotide. Everyone is getting? May I have some responses in the chat box? Any doubts till yet? Yes, students. Okay, good, clear, nice. So let us discuss these now, the structure of a nucleotide. Okay. Nucleotide. So the nucleotide, it consists of three components. Nucleotide is consist of three component. The very first is ribose sugar. The very first component is ribose sugar, which is a pentose sugar. Okay. Ribose is one of the pentose sugar, right? Do you all know sugars? है ना? So you can write inside whenever you required the basics or whatever basics we are taking. So you can write them in margin, right? So as we are discussing here ribose sugar. So ribose is one of the pentose sugar, right? So do you all know that the sugar is the polyhydroxy derivative of aldehyde or ketone, right? Right? The molecule containing multiple OH groups with a functional group carbohydrate or ketone is called as sugar. Okay? है ना? Sugar is the polyhydroxy derivatives of aldehyde or ketone. Polyhydroxy means multiple OH groups with the functional group either aldehyde, that is CHO, or either ketone, C double bond O. That is called as sugar, right? So here in the nucleotide, we have a ribose sugar, which is so sugars they starts from in the nature we have sugars starting from 3 carbons. Okay, so there are 4 carbon sugar, there are 5 carbon sugars, there are 6 carbon sugars. So the ribose is a pentose sugar. What it means that this sugar is consist of 5 carbon. Okay, that's why it is called as pentose sugar. Ribose is one of the pentose sugar. Okay. So ribose is one of the pentose sugar which contains aldehyde as a function group. So it's an aldopentose sugar. Okay. Ribose, it's an aldopentose sugar. Getting this much clear? No confusions? Good. Okay. So ribose is an aldopentose sugar. You can write here. Now, this is an extra knowledge. Actually, the questions are can be being asked on this topics, right? And everyone knows that DNA is consist of deoxyribose sugar and RNA is consist of ribose sugar. है ना? But actually, we should know what is mean by ribose sugar, or ribose is one of the pentose sugar, a 5 carbon sugar, which is an aldehyde type of sugar, right? So ribose is an aldopentose sugar. Now the next component of the nucleotide is phosphate. There is an phosphate group, that is PO4. And the very third component of the nucleotide, so it is consist of 3 components, right? And the third component is nitrogen base. The third component is nitrogen base. So now we are going to draw the structure of each and every component. Okay, so you can draw with me simultaneously. So what is ribose sugar? We called it is a 5 carbon sugar, right? And it is in ring structure. Okay, there is an ring structure of this 5 carbon, right? So this is in carbon number 1, carbon number 2, carbon number 3, carbon number 4, and here to this carbon number 4 here is attached carbon number 5. Okay, so this is the structure of your ribose sugar. Okay, and there is OH and H groups attached. This getting everyone? Okay, just hold on for a second, everyone. Okay, so this is the ribose sugar, which is 5 carbon, and then we have a phosphate group, right? We have a phosphate group which is consist of one phosphate and oxygens, 4 oxygens, right? So this is PO4. Then let's come to the nitrogen bases. So what is nitrogen base? Nitrogen base are nothing but the organic compounds, organic cyclic compounds consist of nitrogen and carbon. Okay, what is this nitrogen is bases? So nitrogen bases, they are nothing but the organic cyclic compounds consist of carbon and nitrogen. These are called as nitrogen bases. Okay, so nitrogen bases, we have two types of nitrogen bases in DNA or in nucleic acid. Okay, both in RNA and DNA. So the first class is purines. First class is purines and the second class is pyrimidines. Now, these purines, they are of two type. These purines are of two type, adenine and guanine. And pyrimidines, we have three types, cytosine, thymine, and uracil. And all these nitrogenous bases, they can be designated by a one letter code. Adenine is designated by A, guanine is designated by capital G, and cytosine by capital C, thymine by capital T, and uracil by capital U. So these are the designation for these nitrogen bases. Okay, so do you know, when we are saying that the DNA is the double helical structure, right? It's a double helical structure consists of two strands, and these strands, they are made up of nucleotides. So now these nucleotides, in among these nucleotides, so this nucleotide is consist of these three component, right? Ribose sugar, phosphate group, and nitrogen bases. So among these three components, the very first two components, that is ribose sugar and phosphate group, they actually constitute the DNA backbone. What they constitute? They constitute the DNA backbone. Okay. And to this backbone, in between, the nitrogenous bases attached. Nitrogen bases, they are responsible for holding two DNA strands together, or the two DNA strands, they are hold it together with the help of nitrogen bases. So see, this is the sugar, right? There is phosphate, then sugar, then phosphate. So such in such a manner, the in such manner the DNA backbone is consisted: sugar, phosphate, sugar, phosphate, sugar, phosphate. And to these sugars, the nitrogenous bases are attached. To this sugar, the nitrogenous bases are attached. Okay, getting this much? Yes, got it? Now we have to actually understand the role of nitrogen bases here and the structure of this nitrogen bases in detail. Ready everyone? Got this much? Yes? Okay, chalo. Yes, okay. I can get the response from Satyam, Tushar, Ganga. Oh, okay, great. Chalo. Soumya. Okay. So now let us understand the role of this nitrogen bases in this. Okay. Now, as I told you, the backbone consists of sugar, phosphate, right? And where these nitrogenous bases are attached, they are attached to your sugar. Okay. So like this, we can have the backbone, right? And to this sugar, the nitrogen bases are going to attach. So now we have adenine, guanine, cytosine, thymine, and uracil. So adenine, guanine, cytosine, and thymine, these four bases, they are present in DNA. And in RNA, we have adenine, guanine, cytosine, and uracil. So RNA, it doesn't contain thymine. Instead of thymine, what it has? Uracil. Okay, so this is the first difference between the DNA and RNA, that the pyrimidine is different. Okay, now these bases, they have fixed pairing. These bases, they have fixed pairing. As we said that the bases are going to hold two strand together because they have ability to pair with each other, right? Bases have ability to pair with each other, and always purine pairs with pyrimidine. Purines pairs with pyrimidine. So adenine, it pairs with thymine, and guanine, it pairs with cytosine. As we know that adenine and guanine, these are purines, and thymine and cytosine, they are pyrimidines. So adenine and thymine, guanine and cytosine, they are going to pair with each other, right? So whenever we talk, okay, this is very important now. uh, Listen it very carefully. So whenever we say that molecules, they are staying together, they are pairing with each other, right? They are holding each other. So it means that there are some forces of attraction in between them, right? There are some forces of attraction in between them. And in the chemistry, the forces of attraction, they are nothing but the bonds, right? So in the chemistry, the forces of attraction, they are nothing but bonds, right? So here the adenine and thymine, they are going to hold each other, right? They are pairing with each other, means they are forming some bonds in between, right? And the type of bonds they are forming in between them is hydrogen bonds. Which type of bonds they are forming? Hydrogen bonds they are forming. So in between the nitrogen bases, there is the presence of hydrogen bond. Which bond? Yes, which bond? Hydrogen bonds. Okay, so hydrogen bonds are the weak bonds. Hydrogen bonds are the weak bonds. Okay, they can be formed and they can be broken easily. Do you know in the chemistry there are two types of bond, strong bonds and weak bonds, right? There are strong bonds and weak bonds, or we say strong bonds and weak interactions as well, right? So hydrogen bond is one of the weak interaction or weak bond, can be formed immediately and can be broken, right? Strong bonds mostly includes the ionic bonds, covalent bonds, and coordinate bonds. Okay, why I'm saying here, because now to understand the sugar phosphate backbone, how this backbone is created, with the help of which bond we will require this. Okay, that's why we are studying this, that in the chemistry there are two types bond, right? Strong bonds and weak bond, right? And the chemistry which we use in the biology is called as biochemistry, right? Yes. So in the biochemistry, we have the strong bonds and weak bonds. Strong bonds includes ionic bonds, covalent bonds, and coordinate bonds, right? And weak bonds includes hydrogen bonds, weak ionic bonds, or we say weak ionic interactions. See, ionic bond is different and weak ionic interaction is somewhat different. Okay, weak ionic interactions, van der Waals forces, and so on. Okay, so hydrogen bonds, they are the weak type of bonds. And you know what? Now there is multiple hydrogen bonds formed in between the purines and pyrimidine. Okay, purine and pyrimidine, they form multiple hydrogen bonds in between them. So the number is fixed. Adenine always forms two hydrogen bonds with thymine, and guanine, it forms three hydrogen bond with cytosine. So this we always have to remember, the pairing adenine and thymine, and ATGC, the pairing is always ATGC. A pairs with T with two hydrogen bonds, and G pairs with C with three hydrogen bonds, right? In case of RNA, they are mostly single stranded, right? There are very rare RNA which could be double stranded, so there will be no pairing, right? So now this is called as, so nitrogen, so two nitrogen bases, right? So these are the nitrogen base. So these nitrogen bases, they are pairing with each other. So now this formed is called as base pair. Okay, this is called as now base pair. Okay, if it is single nitrogen base, it's one base, right? And when it is paired, it is base pair. Okay, so in the double helical structure of the DNA, there are base pairs, right? There are base pairs always, right? Getting this? What is mean by base and what is mean by base pair? Base pair means the paired nucleotides. Not only nitrogen base is there, there will be going to be complete nucleotide. Understood? Yes? Clear? Okay. Now we need to understand one more terminology here, that is nucleoside. Okay. So on this there could be the questions. Okay. What is the difference, means what is mean by nucleotide and what is mean by nucleoside? Okay. Let me clear the board. One more thing I forgot to ask, that everyone is getting my handwriting? Yes? Is there any problem with handwriting? Or you are understanding it? Yes? Okay, great. And if you have any doubts at any point, okay, while I'm teaching, you can add it into the chat box, okay? Okay, so now let us understand the fine difference between the nucleotide and nucleoside. Okay, thank you. No problem with handwriting. Chalo, nice it is. So, nucleosides and nucleotides, right? There are two words, sides and tides. So, this unit of DNA is nucleotide. Unit of DNA is nucleotide. And we can form, yes, was there? Is there any problem? Okay. So, nucleotides and nucleosides. So, as we now, up to now, we are now very much clear that, so in an smaller singular unit in the DNA, there is going to be a 5 carbon sugar, right? And there is going to be a phosphate group, right? There is going to be a phosphate group and a nitrogenous base. So for now, we are going to just draw a square for nitrogen base. Further, we are going to talk about its structure. So among this, anyone could be, right? So to assemble a nucleotide, what we need to do is we need to add one of the nitrogen, right? One of them, right? We need to add one of the base onto the sugar. So now this is very important. इस पे क्वेश्चंस पूछे जाते हैं. ओके, दिस इज वेरी इंपॉर्टेंट दैट on which carbon of sugar the nitrogen base is being attached. Okay, so here if we give the number, this is carbon number 1, this is carbon number 2, carbon number 3, carbon number 4, and here we have carbon number 5. So on the carbon number 1 of the ribose sugar, the nitrogen base is being added. Okay, and in the nitrogen base, we are going to see that the structure of nitrogen base. So, in the nitrogen base, actually, it is The nitrogen base is added to the carbon with the help of nitrogen in it. Okay, so the nitrogen in the nitrogen base is going to bond with the carbon of this ribose sugar. So here, so whenever there is attachment, there is a bond, right? So this bond is called as an glycosidic bond. The bond in between sugar and nitrogen base is called as what? An glycosidic bond. It's an covalent type of bond. It's an covalent type of bond. Getting this is very important. Okay, sometimes the questions are being asked. The nitrogen bases, they are attached to the sugar with the help of which bond? So it's very simple, and glycosidic bond. And now we will see how to remember it. Okay, how to remember it? See, this is what? This is a sugar, right? This is an carbohydrate. It's a sugar. And sugars, they are also called as glyco. Sugar, this word is also called as glyco. So, and this sugar is forming the bond with nitrogen. So that's why, so whenever there is covalent bond between two sugars or between sugar or some other molecule, so that bond is called as glycosidic, है ना? Because there is involvement of a glyco group, right? Sugar group. Okay, here it is sugar group. Okay, so that's why it is called as glycosidic bond. Simple. And this glycosidic bond is the with n atom, right? Nitrogen atom. That's why n glycosidic bond. Okay, so it's more properly one one n glycosidic bond, right? Carbon, it's on carbon number 1, right? So it's one n glycosidic bond. Clear, everyone? Important thing, right? So you can add any of the base here, A, T, G, C. Okay, we can write in, or you can add A, you can add T, one of them, only one of them. Okay, C, like this. Yes. Now the thing is, when there is only sugar and nitrogen base attached to it, that time this complex is called as nucleoside. Okay, that time this complex is called as nucleoside. And when we are going to attach this phosphate group to the nucleoside, when we are going to attach this phosphate group to the nucleoside, then it will become a nucleotide, which is the actual monomer of your nucleic acid. So the phosphate group, it's being attached to the carbon number 3. It's being attached to the carbon number 3 of your sugar. Understood? On the carbon number 1, nitrogen is base, and on the carbon number 3, phosphate is going to attach. You know what? To this carbon number 3, there is an OH group here. Okay. All these carbon are further attached with OH and H groups, right? Yes. So, so by just, so this phosphate group, it reacts with the OH group, and here is the addition of the phosphate group giving. Okay. Understood? Yes, clear? And glycosidic bond, where the nitrogen base is going to attach, what is nucleoside and what is nucleotide? So here could be the question being asked, what is nucleoside or what is nucleotide? So whenever I'm saying, uh, here there is possibility of being question asked, so mark it IMP. Okay, underline that things. Clear this much? Okay, now let us go and talk about the detailed structure of this nitrogen bases, how exactly they look. Ready, everyone? Can we go ahead, or want any uh, repeat explanation of anything? Okay, so now let us discuss about the nitrogen base structures, and then we are going to talk about the phosphodiester bond as well. Okay, so let's go ahead. So here are the structures of nitrogen bases. So Nitrogen bases, we have two types, right? Two types of nitrogen bases we have. One is purine and another one is pyrimidine. So purines are the nine membered ring. Nine membered. Purines are what? Nine membered double ring structures. Purines are nine membered double ring nitrogen bases, right? Or we say structure, right? Can you see the nine members over here in the in the purines, adenine and guanine, right? How the pyrimidines are? They are six membered rings and they have only single ring, right? So both of these are the ring structures, right? Cyclic structures. Purines, they are nine membered, and pyrimidines, they are six membered, right? Now, so this is an parent structure of purine. So this is the parent purine. This is what? This is the parent purine. Nine member structure, right? With four nitrogens. You can see here in the adenine, there are four nitrogens, right? Among these nine members, there are four nitrogens and rest of them are carbon. So you can give numbering from here. 1, 2, 3, 4, 5, 6, okay, and this is 7, 8, 9. Okay, so the numbering to the purines is given in such a manner. So you can see the adenine is, you can see, so see, this is the parent purine, right? This is the parent purine. Carbon number 3 of pentose sugar. So further we are going to discuss about that. It is going to give rise to phosphodiester bond between two nucleotides. Okay. It's a simple covalent bond. Okay. It's a simple covalent bond between carbon number 3 and phosphate. Okay. And further this phosphate is going to attach with the next nucleotide that will give us phosphodiester bond. Okay. Yes. Yes. Sashwati, right? Sashwati, clear beta? So the bond between carbon number 3 and pentose sugar. Carbon number 3 of pentose sugar and phosphate is in simple covalent bond, and further this will help us to form the phosphodiester bond in between two nucleotides. Clear? Okay, great. Chalo. ठीक है, ठीक है. Yes. And the bond between carbon number 1 and nitrogen base is N-glycosidic bond. Okay? Yeah. So see, now try to understand here. The purines, they are the nine membered ring structures, right? So we have numbered the nine members in the purines. So this is an parent structure, right? You can see in the both the case, this is an parent structure and modification in this parent structure leading to the variant type of purines, that is adenine and guanine. So if this is an parent structure, whatever I have drawn here is, if it is a parent structure, so I can write it as purine. I can write it as purine. So in the chemistry, the double ring structure of nitrogen, they are named as purines. Okay. And single ring structures of nitrogen, they are named as pyrimidines. So purines. So if we discuss about the first very purine, if we write the chemical name of the first very purine, see, on the fourth position, you can see there is an amino group, right? There is an amino group. So we can write it as 4-amino purine, right? So the adenine is nothing but chemically 4-amino purine. Okay? The adenine is 4-amino purine and the guanine is, you can give numbering again, 1, 2, 3, 4, 5, 6, 7, 8, 9, right? So what is guanine? Guanine is actually 2 amino 4 oxy purine. Understood? So adenine is 4 amino purine and guanine is 2 amino 4 oxy purine. Okay? Understood? So this can also be asked. What is adenine? You should know it is 4 amino purine. Okay? Or guanine. So let's discuss about the pyrimidines. Okay? Now pyrimidines, they are six membered rings, right? Six membered ring structures, and you can also give the numbering to this, right? So numbering to this also given in the anti-clockwise manner, and you can start from the bottom one nitrogen. Okay, 1 2 3 4 5 6. Okay. Now here we can see, we can give the numbering to all the nitrogen bases, right? 1 2 3 4 5 6, right? And here it is 1 2 3 4 5 6. This much clear? Yes, everyone? Okay, just everyone just hold on for a minute. I'll be back. 1 minute, please. Audible? Am I audible? Okay. Chalo, so let's go ahead. So these are the structures of your thymine, cytosine, and uracil. So you can draw the structures. And now let us talk about the chemical names of this thymine, cytosine, and uracil that will help you to remember the structures of these nitrogen bases. Okay. So we will start with the cytosine. So the numbering to the pyrimidines will start from this carbon. Okay. So here this carbon will be your 1 number carbon, and you have to give numbering in anti-clockwise direction. And here numbering, see there is rule. Do you remember in the chemistry? We always give numbering in a such a way that all the branch group, right? The branched points or the attached group, they should get the minimum numbering, right? They should get the minimum numbers, or the sum of branch point should be least. So in such a way, we are going to number it. So the anti-clockwise numbering will be the right numbering for this. Okay, so here it will be 2, 3, 4, 5 and 6, right? So according to this, if you now name this cytosine, right? According to this, if you now name this cytosine, what is it? At the fourth position, it has an amino group, right? At the fourth position, it has amino group. So it is 4 amino 2 oxy pyrimidine, right? It is 4 amino 2 oxy pyrimidine, right? So now if we talk about the uracil, what is uracil? It is 1, 2, 3, 4, 5, 6. It is 2,4-dioxypyrimidine. Noted everyone? It will be 2,4-dioxypyrimidine. Done? This much clear? Now see an interesting fact here. If you compare the structure of uracil with the structure of thymine, everything is same, right? The structure of uracil and the structure of thymine, everything is same. Only there is one more methyl group, CH3 group is added onto the fifth carbon of, है ना, fifth carbon. It is making it as thymine. So instead of writing a big chemical name, we can write it as 5-methyl uracil. So what is thymine? Thymine is 5-methyl uracil, right? What is cytosine? 4-amino 2-oxypyrimidine. What is uracil? 2,4-dioxypyrimidine. And what is thymine? So you can say thymine is 5-methyl uracil. So you can easily modify uracil into thymine, right? So modification of uracil into thymine is very easy. You have to just add a methyl group. Clear this much? Clear all about the structure of nitrogen bases? Yes. So now we are going to talk about the packaging of the DNA. Now we know how DNA is formed. We are just going to talk about the phosphodiester bond formation and then further the packaging of DNA. Okay, great. Chalo. So let's go ahead. Now we are clear with the structures of nitrogen bases, and now let us talk about the phosphodiester bond. Okay, chalo. So see here. What is going to happen then? Now, here is a sugar molecule. To its carbon number 1, there will be a nitrogen base. So we are going to just denote it by the letter, respective letter. Like suppose it is adenine. So A. So on the carbon number 3, we have the phosphate group attached, right? So in a such a way, see, you can see here the carbon number 5 is free. Now one more thing we need to understand is, see, we have given the numbering to the nitrogen bases, right? The atoms present in the nitrogen bases, we have given numbering as 1, 2, 3, 4, 5, 6 and so on, right? So now in this complete nucleotide, we have two such ring structures, right? And to both the ring structures we have to number, right? So to avoid confusions in between numbers, if I am saying the carbon number 1, so which carbon number is it, of nitrogen base or it is of sugar? So to avoid this, we number sugar molecule not as carbon number 1, 2, 3, 4, 5, but we number them as prime. Okay, we just put a dash on the head of the number, that is called as prime. So we give, it is read as prime. Okay, so we give numbering as 1 prime, 2 prime, right? 3 prime, 4 prime and 5 prime. Okay. So here to 1 prime position there is the attachment of nitrogen base, and at the 3 prime position there is the attachment of phosphate group, right? So now this phosphate group is further going to form a bond. So if now next upcoming nucleotide, let us draw the next upcoming nucleotide. So that will be again consist of a nitrogen base, and the we are just drawing the phosphate group like this. So the 5 prime carbon, the 5 prime carbon of, see here, the 3 prime to the 3 prime there is phosphate group, and in the upcoming nucleotide the 5 prime carbon is free. So now this 5 prime carbon is going to react with this phosphate group, right? So further, this is how the upcoming further nucleotides are being attached. So the bond formed between the 3 prime carbon and the phosphate and the 5 prime carbon of the sugar is called as phosphodiester bond. Is called as what? Phosphodiester bond. Because we can see here two oxygens, they are involved in bond formation. So that is phosphodiester bond. What it is? Phosphodiester bond. There is one phosphate group and two oxygens, they are involved in this covalent bond. So this complete bond is called as phosphodiester bond. So the bond between nucleotide is phosphodiester bond. So now can we say that the DNA backbone is consist of sugar and phosphate with the help of phosphodiester bond in between, right? And the nitrogen base, they are attached to the sugar phosphate backbone with the help of N-glycosidic bond, right? And now, do you know one more thing, important thing about the DNA is that, so see, when we are continuously joining these nucleotide with each other, so at the top we can see the 5 prime carbon of sugar is free. So can we say this end of the strand as 5 prime end, right? And if we will go if we'll continuously adding the nucleotides, but at the end we can see the 3 prime OH group will be free. At the end we can see the 3 prime, if there is no phosphate, है ना? If there is no phosphate in this nucleotide, there will be the 3 prime OH group free. So the another end of the DNA strand is called as 3 prime OH. Okay, 3 prime, sorry, 3 prime phosphate, right? So at the top there is 5 prime OH group is free, and at the bottom there is 3 prime phosphate group is free, right? So this is a designation for the DNA strand, 5' OH and 3' phosphate. Okay, got it? So, always remember the two strands of the DNA, they are complementary to each other, they are in opposite direction. So, the another strand is being synthesized in opposite direction. So, in opposite to 5' OH, it will be having 3' phosphate, and in opposite to 3' phosphate, it will be having 5' OH group free. Understood? So this nature of the DNA, that two strands of the DNA, they are running in an opposite direction. So, and now in between, right? Now in between, what is going to be there? Nitrogen bases like adenine, guanine, thymine, right? Cytosine, guanine, like this. And these will be base paired with their complementary nitrogen base like thymine, cytosine, with the help of hydrogen bonds, right? GGC. So now these nitrogen bases, they are always complementary to each other, right? The base pairing is fixed. So this fixed base pairing is called as Wobble base pairing. This is called as what? Wobble base pairing, which is fixed. Adenine will always pair with thymine, and guanine will always pair with cytosine with 2 and 3 hydrogen bonds respectively. So now these two strands, they are called as complementary strands. They are called as what? Complementary strand, because they base pair with the complementary bases and they are opposite in direction. So how the DNA strands are? They are complementary, right? So that's why we say the DNA is double helical structure of two complementary strands. Understood? Understood? The 5' OH and the 3' 5' OH and 3' phosphate group. Yes? Clear this much? So one end of the DNA is 5' and another end of the DNA is 3', right? Okay. Now let us try to understand how this DNA, they are packed and kept in our cell, right? So let us once revise what we have learnt up to now, and then we'll go further. So what we learnt up to now, there are two types of cells, prokaryotic and eukaryotic. What's the difference in between? The difference in between is what? Prokaryotic cell, they lack true nucleus. Eukaryotic cells, they have true nucleus, right? And further we learned that The organisms we divide into two type on the basis of what? On the basis of prokaryotic and eukaryotic cell type. Prokaryotes and eukaryotes. And again on the basis of the number of cells they have, unicellular and multicellular. So on the basis of this, with some features, we have classified them into the five kingdoms. And then we got to know that the DNA is the genetic material, and then we learnt about the constituents of the DNA, that is nucleotide. So nucleotide is formed by the one nitrogen base and the phosphate group attached, right? One nitrogen base and phosphate group attached. So this is how the nitrogen bases are going to be. At carbon number 1 the phosphate, and at carbon number 5, at carbon number 1 the nitrogen bases, and at carbon number 1 the phosphate groups. So now this upcoming nucleotide from this 5 carbon is going to attach to the 3' OH of your preceding nucleotide, right? So this bond is phosphodiester bond. This end here 5' end, and this 3' OH end. Okay. So one end of the DNA is 5', another end of the DNA is 3'. At the 3' end, you will get the free OH group, and at the 5' end, you will get the free phosphate group. Okay. Phosphate group is attached where? It is attached to the carbon number 5. Nitrogen base is attached where? It is attached to the carbon number 1. So all the upcoming nucleotide, they will be like this: carbon number 1 nitrogen base, and at the carbon number 5, okay, at the carbon number 5 there is phosphate group. So now this phosphate group is going to bond with their carbon number 3 of preceding nucleotide, right? So always at upcoming nucleotide will be with the phosphate at carbon number 5 and 3' free OH group, and it will go and again bind to the previous 3' free OH. So like this we will get the two strand 5' phosphate and 3' OH free at the end, and the next strand will be opposite to it. It will be having opposite to it 3' OH group free and 5' phosphate group free. Okay, understood? So now see, if we talk about the direction of DNA synthesis, if we talk about the direction of DNA synthesis, so DNA is always being synthesized in 5' to 3' direction. As we can see this, the first nucleotide is going to be like this. Okay, phosphate at carbon number 5 and nitrogen base at carbon number 1. So 3' OH group is free, right? So this is 5' group which is free, and to the 3' the upcoming nucleotide is going to attach. So what is the direction? 5' to 3'. The DNA is being synthesized in which direction? 5' to 3' direction, right? Because at the end you will get the free 3' OH group, that is called as hydroxyl group, right? So on this also the questions are being asked. The direction of synthesis of DNA strand, it's always 5' to 3' direction. In case of RNA also it is 5' to 3' direction. Okay. What will be the only difference between the synthesis of DNA and RNA? The sugar. The difference in between what? Sugar and the bases. Now let us discuss the second difference in between the DNA and RNA. What is the second difference in between the DNA and RNA? That is sugar. So the sugar present in the DNA is deoxy sugar. Okay, it means what? At the 3' position, it has OH group, and at the 2' position, it has only H group. But in case of ribose sugar, at the 3' position, there is OH group, and at the 2' position also there is OH group. At the 2' position also there is OH group. That's why it is called as ribose sugar, but in the DNA it is called as deoxyribose. Why? Because there is one oxygen less at 2 prime position. Which position? 2 prime position. Okay, which is making the DNA more stable, right? What is the reason behind it? That nature has converted. In the nature, first there was RNA formation. In the nature, first there is the formation of RNA, ribonucleic acid. But in the ribonucleic acid, there are two oxygen group free, right? 3 prime OH and 2 prime OH. So the 3 prime OH was going to react with the 5 prime phosphate, right? The 3 prime OH was going to react with the 5 prime oxide. So it is now in the bond, so it will not react. But the 2 prime OH, it's free, right? You can see here the 2 prime OH, it is free. So there are more chances that this 2 prime OH can undergo reaction with some other molecules readily. That's why RNA is more interactive than DNA. But this is not suitable to store the genetic information. Genetic information should be always stored very safely, right? The genetic information should be always stored very safely, right? So to make molecule more stable, this is making the RNA molecule unstable because it can readily undergo reaction with any molecule because of the presence of O. Oxygen is highly reactive, right? So to make it more stable, the nature has removed the oxygen from 2 prime position, so leaving the deoxy sugar, and that deoxy sugar we used in DNA. Clear everyone? Direction of DNA synthesis, nucleotide, how the nucleotide is being attached. So this is also very important. We have at 5 prime end free phosphate group, at 3 prime end we have hydroxyl group, right? How a nucleotide molecule is consist of? It consists of a sugar, and to this sugar add carbon number 1 nitrogen and add carbon number 5, right? At carbon number 5 there is phosphate. This much clear? Yes, Pakka. You have to remember these two carbon numbers. At carbon number 1, nitrogen base. At carbon number 5, there is phosphate group. Three carbon OHs free to bond with upcoming 5 prime phosphate, right? Upcoming 5 prime phosphate. Chalo. So now we are going to first discuss about the organization of the chromatin in the eukaryotic cell. Okay, what we are going to discuss about organization of chromatin in eukaryotic cell. Then we'll discuss about the prokaryotic cell. Okay, so in case of eukaryotic cell, so we know that eukaryotic cell is a well organized cell with a nucleus, and in nucleus there is genetic material. So as we know that the DNA is quite longer structure, right? So to keep it inside a small nucleus, it need to be packed, right? So DNA, the double helical structure is packed into a very small structure, right? So we are going to condense it. Condense it means compactly pack it. Okay. Let us give a small example. If we have a thread, right? If we have a very long thread, storing it is very difficult. But if we are wrapping it around a bundle, right? If we are wrapping it around any object and making its bundle, so storing a very long thread is easy in very small space, right? So similarly, it's going to be. No, beta. Purnima, what you are saying? So what you are saying, Purnima, to sugar we have to give anti-clockwise numbering. What you are saying, beta? Two sugars? No, we'll not get the five numbering. See, I'm saying I'll show you again. Okay. So you can see here. This my cursor. Okay, let us discuss again. So if this is your sugar, right? This is carbon. This is a carbon. This is a carbon. And this is a carbon. Okay, actually the five carbons in the sugar, they are not in ring. Only four carbons are in ring. Okay, one of the carbon is out of ring. One, two, three, four, five, right? And at this fifth carbon, to this fifth carbon, the phosphate group is attached. Phosphate group is attached to the fifth carbon. Okay, like this.
Yes, yes, ma'am. Clear.
Yes, like this. Okay.
Done. Yes, ma'am.
And here to the one carbon, there will be the nitrogen base, right? Any one of the adenine, guanine, cytosine, or thymine. So this is a nucleotide, right? Here will be the OH group. Here will be the H group. Here is H. Here is H also. Okay. Here is also H. Okay. Getting?
Yes,
Yes.
yes, ma'am.
Okay. Here is one more H. Okay. Clear, everyone? This you have to remember. This is a nucleotide, right? This is a complete nucleotide. At carbon number 5, the phosphate group. At carbon number 1, the nitrogen base. And the 3' OH is free to receive the upcoming phosphate from the upcoming nucleotide, right? है ना? The next nucleotide will be same. That will be having the 5' phosphate. So that 5' phosphate is going to react with the 3' hydroxyl group. Okay? Clear? Can we go ahead?
Yes, ma'am.
Okay, good. Chalo. Yes, you can respond me with audio. It will be more better, है ना? To be interactive and understand. Chalo. So let's go ahead. Let's talk about the packaging of the DNA now. So as we know that DNA is a double helical structure. So as we was discussing that if there is a long thread, so it will require so much space, right? But if we will wound it around in any object, so it can be stored in very small space, right? So similarly is done with the DNA. So DNA is very long, right? So we are going to pack it, wrap it around some proteins. We are going to wrap it around some proteins. So these proteins, they are called as histone proteins. They are called as what? Histone protein. So we use
This conference will now be recorded.
So we're discussing the packaging of DNA, right? So DNA, they are wrapped around the histone protein. They are wrapped around which protein? They are wrapped around the histone proteins. So to wrap the DNA into the compact structure, we use 5 types of histone protein. How many types of histone protein? 5 types of histone protein, and they are termed as H1, H2A, H2B, H3, and H4. So, what are the names of histone protein? H1, H2A, H2B, H3, and H4. Okay, so these are the 5 proteins we use to pack the DNA. So, now what is chromatin actually? So, the chromatin is the compact structure of DNA plus histone protein. DNA plus histone proteins. So, in packaging of the DNA, we have different steps. So, am I not audible? Am I or not? Am I audible? Hello?
Well, you can
Yes?
continue. You're audible.
Okay, okay. Thank you. Okay. So we are using these five histone protein. Okay, we are going to use this 5 histone protein to wrap the DNA molecule. So the DNA molecule is actually wrapped around an histone octamer. The DNA molecule is wrapped around on histone octamer. Histone octamer. So what is meant by octamer? Mer means single units, right? Mer means single units. How many units, single units are there? Octa. Octa means 8, right? So there are 8 units of like this, okay? There are 8 units of histone protein together form a bead-like structure, and on this bead-like structure the DNA is wrapped. From this on this bead-like structure, the DNA is wrapped, right? And to link this bead-like structure, one histone is utilized, that is H1. Okay. So to form histone octamer, we are going to utilize H2A, H2B, H3, and H4. So these 4 units or these 4 proteins going to be the part of histone octamer, and the H1 is called as linker histone. H1 is called as what? Linker histone. So when the DNA is being wrapped around the histone protein, so it looks like a string on bead structure. Okay, so the histone octamers, they are looking like beads, and on these beads we are wrapping over the DNA like this. So this structure form is actually called as beads on string structure, okay, which is further called as nucleosome, which is further called as nucleosomes. So now these nucleosomes, so first there was a DNA double helix, right? Then we have wrapped this DNA double helix around histone, giving rise to nucleosomes. Now this bead on string structure or this nucleosome is compactly packed with each other. Okay, so this structure is now condensed. This structure is now Condense.